Bien, alors Alain me donne maintenant le privilège de présenter notre deuxième conférencière. C'est Rebecca, professeure Rebecca North, une Canadienne de l'Ontario. J'ai appris aujourd'hui qu'elle a terminé donc ses études de baccalauréat donc en limnologie. Ça c'était en fait son PhD pardon à l'Université de Waterloo en 2008 et après elle a fait son postdoc à Trent, l'Université de 2008 en 2012. Alors Rebecca s'est toujours intéressée, donc elle est maintenant professeure, disons assistant professeur donc à l'Université de Missouri où elle s'occupe donc de tous les aspects reliés à la qualité de l'eau. Et elle s'est toujours intéressée à tout ce qui est problème d'eau, des eaux douces, l'impact donc par exemple de l'agriculture sur les eaux douces. On connaît tous les problèmes d'eutrophication donc des lacs, des rivières ici, dû entre autres à tous les aspects par exemple de fertilisation, de surfertilisation, si vous me permettez l'expression, de nos champs agricoles. Alors tous ces éléments-là, le phosphore, l'azote se ramassent dans les eaux et tuent finalement la vie aquatique des lacs. Et évidemment, tout cela est amplifié aussi dans le contexte de changement climatique qu'on connaît aujourd'hui. Et aujourd'hui, donc, elle va nous présenter sa recherche à l'Université de Missouri, où, comme si j'ai bien compris, c'est une étoile filante. Donc, elle a commencé sa carrière là-bas. Et elle est là pour donc développer cette filière-là donc de recherche sur la qualité de l'eau. Et la présentation qu'elle va nous faire aujourd'hui, c'est sur Water Resources in a Changing Climate. Mais avant, j'aimerais souligner aussi qu'elle est membre de plusieurs associations, entre autres l'Association américaine de limnologie et d'océanographie, membre de la Société phycologique des États-Unis. Elle est aussi membre de l'Association internationale pour la recherche. On a le groupe de recherche sur le Grand Lac. Et aussi elle est membre donc de limnologie, de la Société canadienne de limnologie. Donc sans plus perdre beaucoup de temps pour la présentation, je vais passer la parole à professeur Rebecca qui va nous parler donc de Water Resources in a Changing Climate.
Thank you very much. I think I'm good here. So I'll follow Dr. Joseph's lead and start by introducing you remotely to the other water faculty in the School of Natural Resources at the University of Missouri. So as Dr. Joseph referred to, we've had some new hires. We've hired 4 new faculty in the area of water resources, and we have complementary skills. And so we're building this program to be able to address issues in water resources in a changing climate. So as mentioned, I caught some of that. I'm a lake ecologist, and so, well, I used to be a lake ecologist. Now I should call myself a reservoir ecologist, because in Missouri, all of our water bodies are man-made. I co-lead the University of Missouri Limnology Lab with a stream ecologist, Dr. Alba Agarich. We've also recently did some joint hires with engineering, and so this has brought to the team Noah Locius. He's a water resources modeler and kind of human resources management component. And then Dr. Ranjith Adawada has been working a lot in the agroforestry regime in terms of watershed management. And so I'd like to think that all of these different specialties are brought together by our newest faculty member, Damon Hall. So he is a social scientist and works in water resources policy and planning. And so hopefully this new team that has been formed will be able to do some great integrative work moving forward. So I'll start by giving a few slides on each of us to kind of give you a background and hopefully establish links for future collaborations. So I am a limnologist. My work revolves around 4 different themes. I look at eutrophication, or nutrient inputs into water systems and how it affects water quality. I also look at landscape modification, so kind of converting more kind of pristine or forested landscapes into more urban or agricultural settings and the impact that has on sediment load and nutrient dynamics. I've also done some work on aquatic invasive species, and as you're familiar here, invasive dracenids or zebra mussels can have huge impacts in terms of water quality. And so lucky for me, when I got the position in Missouri, they have, zebra mussels have invaded down there as well. So it's a continuing theme. And I also look at climate change, so both kind of the hot and the cold effects of climate change on aquatic ecosystems. And so you're probably familiar with this image. This is showing a satellite image of the western basin of Lake Erie. This is the harmful algal bloom that resulted in the city of Toledo being without a source of drinking water for 500,000 people over a course of 3 days. So a lot of my work revolves around trying to understand why we're seeing this expansion on a global basis of these harmful algal blooms. I was fortunate enough to walk into a position to inherit some long-term research programs. So we have 2 long-term running programs. The first is the statewide lake assessment program. And so these are funded through the Environmental Protection Agency and administered through the state government. This has been continuous data collection on water quality parameters from 1978. So it includes about 29 summers of monitoring, and we cover about 253 lakes. And again, here you'll have to forgive my use of the word lakes, because these are all, in fact, reservoirs. So we sample a couple of times during the summer, and this is samples collected mostly by undergraduate students during the summer months, and then the samples are analyzed in our lab. This is complemented by a citizen science program. And I did bring some a recent report, so if anybody's interested, I have a few of these around. This has been another long-term running program, so it's gone from 1992 to present, so 25 seasons of monitoring, and it's included 111 lakes, and we have multiple sites on some of these lakes. And so it turns out that retirees have more time than undergraduate students, and so out of them, we get more data. So we have a larger seasonal window, and we get 8 samples per season. So they start a little earlier, they start around April, and go through until September. And so this is, kind of your basic water quality parameters, and able to look at these long-term trends. I also have some graduate students, and one of my students, Erin Petty, is looking at nitrogen versus phosphorus deficiency in phytoplankton communities. And so there's been some recent debate in the literature of whether it's important to control for nitrogen or phosphorus or both. So this is a paper by Hans Perl, It Takes 2 to Tango. Clearly he's promoting the nitrogen and phosphorus. And so she's looking at land use in the watershed. So again, looking at kind of agricultural and urban and forested watersheds and seeing what their contribution to both nutrients and sediments are. We're assessing the response of the receiving water bodies on this in terms of nutrient bioavailability and light attenuation. So the reservoirs in Missouri are primarily brown in color. We have really, really high sediment loads as a result of a lot of agricultural land use. And so kind of looking at how those interactions result in terms of phytoplankton productivity and biomass. I have another graduate student who's looking at light. And basically looking at 2 different sides of the spectrum. So how changes in light affect phytoplankton in our reservoirs. And so we know that dracenids or zebra mussels are filter feeders, so they tend to clear the water column and improve the light conditions. So he's looking at, again, these long-term data sets of before and after to see their impact. And he's also doing some biogeoengineering components. So adding something to lakes, in this case we're adding glacial rock flour, to diminish the light environment. And so if we turn off the lights, can we see, decreases in these harmful algal blooms and really eutrophic systems? I'm also interested in year-round dynamics. And so this is maybe, my Canadian content where I'm like, winter's a thing and it happens. And so I've got 2 students now looking at year-round processes in reservoirs. So what happens in reservoirs when nobody's looking? We tend to have this summer-centric philosophy, probably mostly based around undergraduate availability. But, we drink the water out of our drinking water reservoirs on a year-round basis. And so it's important to understand the dynamics of these harmful algal blooms and the potential to produce toxins on a year-round basis. And so we also know that the conditions that used to kind of diminish the growth of cyanobacteria, such as winter and snow and ice, are declining. And so these changes in terms of winter dynamics may be having an impact and may be related to what we're seeing in terms of this expansion of harmful algal blooms. Also, I'm starting to learn as a new faculty member that you can't be in all places at all times. And so in order to collect this year-round, weekly resolved data, we're employing high school students. And so we just started a new project called Reservoir Observer Student Scientists, or ROS for short. And so these students at a local high school are going out and collecting water samples on a weekly basis to look for harmful algal blooms, algal toxins, and water quality parameters. So I've been doing this for a year now on a local reservoir in the city I live in Columbia, Missouri. And we just got funding to expand this out to an additional 5 states in the Midwest. So I'm hoping to be able to use this gradient, this latitudinal gradient, from Wisconsin and Minnesota, where they have ice cover very similar to here, down to Missouri, where ice cover is pretty intermittent, and see if we see differences in terms of harmful algal blooms. As I mentioned, my colleague is Dr. Alba Agrich. She's a stream ecologist, and she looks at nutrient transport, transformation, and fate, and primarily focused on, again, carbon, nitrogen, and phosphorus dynamics. She studies streams at a variety of different scales. So she's done work on the continental scale, catchment, reach, and morphological units. And her work is very integrative. She uses both field observations and experimental manipulation, as well as lab experiments and modeling to be able to answer some of her questions. And her point here is that some small streams are everywhere, and so we often don't recognize or pay attention to them, but they're diverse and they're spread all over the place and they're important. We know here that what we know about these small streams is very little. So only 21% of all the streams in the US have actually been assessed. So we don't know enough, and of what we do know, about half of those streams are considered to be impaired. And so it's important to understand these small scale processes. But one of the problems with some of this stream work is what's our baseline data, right? So what's a pristine or healthy system? And so here she did a study where she looked at trends in nitrate concentrations in long-term data sets of what we consider to be kind of reference streams. And so the red colors are indicating increasing trends and the blue colors are decreasing trends. And so you can see that in some cases, these unimpacted reference streams are seeing significant increases in nitrate concentrations. So these are some of the questions she's currently pursuing. She also does some kind of manipulative applied research, and so here she's looking at using vegetation, so rooted macrophytes, to be able to improve stream water quality. And she's got a PhD student working on that over in Spain as we speak. She's also looking at the impact of flooding, right? So when streams can be very noisy, and you can see that as you have different water levels in terms of how often things flood and how long they're flooded for, this can have impacts on nutrient spiraling, and specifically, again, carbon, nitrogen, and phosphorus. So she's also looking at this role of the riparian zone on the downstream movement of nutrients. Some of her most recent work, conducted by an undergraduate student at the University of Missouri, is looking at gravel bars. So we often put in gravel bars for fish habitat or to reduce erosion, but they also have biogeochemical consequences. And so she's looking at kind of upstream, downstream effects of putting in these gravel bars. So I mentioned part of the team is also a modeler. So this is Dr. Noel Aloysius, and he has a very challenging task of trying to integrate human and natural systems and make predictions about the future. And so one of his current challenges is to try to deal with this Gulf hypoxia. And so the Mississippi River Basin is this giant river basin you can see depicted here, and composed of both cities and farms. And the goal to be able to reduce hypoxia in the Gulf of Mexico is that by 2035, they want to reduce pollution load, and in this case, they're considering both nitrogen and phosphorus by 40%. And so this is a pretty, I see some cringing, it's a pretty impossible task. And so he's using his skills in modeling to be able to see what we need to do to get there and how realistic that target is. So he's looking at coevolution of human and natural systems. So he considers the kind of social human element as well. And as Dr. Joseph was explaining, if we don't have that human uptake or support of these systems, then it's not going to be effective. So he's looking at the movement of water and pollution controlled by multiple stressors. And he postulates that the problem of sustainability is global, but there are some local solutions that we can apply to achieve our objectives. And so they should account for all of these integrative, iterative kind of components, including the environment, social, and economic influences in space and time. And so this is just kind of a sustainable problem-solving framework that he's developed, where he's trying to work with people to get towards this new state system to be able to achieve these goals of nutrient reduction. Some of his current projects that he's doing is to identify key drivers of diffuse pollution transport in managed river basins, and so he's looking at large river systems in this case. Also evaluating the long-term accumulation and release of both nitrogen and phosphorus in these human-dominated watersheds. He's also evaluating the sustainability challenges in agricultural water management in the tropics. So he does a lot of work in the kind of the Congo River Basin area of Africa. And he's also looking at identifying the impacts of fertilizer runoff and pesticide exposure on wetland vegetation. So a diversity of projects, and he's just been in his position for just over a year now. So he's just ramping up on these things. The other project that Dr. Jose referred to is work done by Ranjith Adawada through the Center of Agroforestry. And so here they're looking at this, what we call MRBI, or Mississippi River Basin Healthy Watersheds Initiative, trying to, deal with questions that came up earlier in terms of water quality benefits of agroforestry. We're very fortunate at the University of Missouri. The university owns 17 experimental farms all around the state, and they're located in different places with different kind of agricultural systems. And so the one he's working on is kind of in the northern part of the state called the Greenlee Center, and they're using a paired watershed approach. And so they have these watersheds in place. They've installed contour strips, which are shown by the gray contour lines there. Some of them, they've applied some agroforestry measures, and then, of course, they have the control. So if they implement these designs, what do they see in terms of improvement of water quality? They're also looking, in addition to nutrients, soil erosion loss. And so here he's showing the management in trees treatments. So if you use grazing management versus row crop management, what impact are you seeing in terms of the effect of implementing these type of techniques? And so it's the percent reduction, and so you can see they're very effective in reducing both sediment, total nitrogen, and total phosphorus from getting off the watershed. He also does some work on landscape position and buffer effects on groundwater nitrogen. So, we've heard some stories out of Iowa and things like that with really high nitrogen concentrations, which is affecting drinking water sources. And so he's looking at whether or not we can use some of these agroforestry practices to be able to minimize the nitrate concentrations in the water. And so again, he's working on a different agricultural research site that the University of Missouri owns, where he's installed wells on a slope. And you can see in the bottom two, they've applied agroforestry buffer and then a grass buffer. And so when you look at the concentrations of nitrate and total nitrogen, you can see at the top of the slope, there was pretty high concentrations at the bottom of the wells and the groundwater. But after the application of this agroforestry buffer, you can see those significant declines in concentrations and similar declines when you put in grass buffers. So these, again, seem to be really effective in terms of reducing nitrate. And he's putting all of this variety of different experimental data together, as well as kind of management considerations and kind of larger data sets such as precipitation, to try to model them in terms of APEX is the model that he's choosing to use in this case. So trying to look at all of these different components, integrate them, and again, make predictions for the future. We're very fortunate, in some ways, in Missouri to have a variety of land uses. And so we have a lot of agriculture in the northern part of the state and in the southern, what we call the boot heel, because it looks like a boot heel. And in the middle, we have our Ozark region, which is primarily forested. So looking across this gradient of land use, he's be able to put in recording and flow measurement systems to be able to get evidence of the impact of this different types of land use. And so here he's taking these variety of different sites in the state, using APEX and SWOT to kind of scale them up and apply different practice techniques to make predictions in terms of the future, in terms of water quality management. And so we're able to kind of get continuous feeds from this data up to satellites. This is an image of our University of Missouri campus, so he can sit in his cozy office and look at data and then make predictions that can hopefully be used as decision tools in terms of management techniques. And finally, our last and most newest member of the Water Resources team is Damon Hall. So as I mentioned, Damon is our social scientist. He works on social and policy research, and he always starts his talks off by including this quote from Jack Thomas, that managing natural resources is 5% managing nature and 95% managing people. And so with that in the back of his mind, this is how he carries out all of his research projects. And so he wants to know how decision makers conceptualize these social ecological systems and how this can be translated into policy and management. And so he spends a lot of time talking to people and engaging stakeholders to build these large qualitative data sets for in-depth analysis to improve baseline assumptions of socio-economical decisions for decision making. And one of the great case studies that he's doing is he's using crowdsourcing or citizen science to be able to get people to measure gauge data, so looking at how much water is in the rivers. And so this is work he's done up on the Boyne River in Michigan, and so they install gauges with water temperatures and these signs, and they're getting people to text in what they're reading on these things, and this is forming these data sets in places where we don't have gauges. So in the States, their USGS installs gauges, but not everywhere, and certainly not all the time, and so this can complement and supplement those data sets. And so he's been very successful in doing that. And part of that success is bringing in the crowd and stakeholders at many steps of the process, not just the end. So he starts off by talking to people in terms of what they need and what they're interested in, and then why they participated. So trying to understand how to engage these people in these processes. And if you want to learn more about what he does, there's his email address there. And also, Dr. Jose and I can obviously put you in contact with all of these people if you're interested. All right, so that was my pitch for the School of Natural Resources, and now I'll move on to talking about winter limnology. And so it's funny, because Dr. Jose and I did not compare our talks before we came today, but we're both going to talk about how to grow plants with light nutrients. And in my case, I'm going to talk about how they are not dormant under the ice in winter. And so my training in limnology, the definition, on the first page of my course notes, was the study of lakes located within 60 miles of a major university campus, but not during winter. When you look at the literature and what we understand about lakes, this is a lot of the basis, and it's pretty much a practical decision on that. And so with this in mind, we've designed a lot of our academic research programs and government monitoring programs with this kind of summer-centric philosophy. It's allowed us to develop some of these traditional paradigms. So this is showing year-round, so on a monthly basis, mean chlorophyll A concentrations. And so chlorophyll A is our proxy for phytoplankton biomass. It's the pigment that phytoplankton produce to be able to capture light. And so we can see here that we expect in temperate lakes to have this nice bimodal curve. And so we designed most of our sampling programs to capture these two peaks, which we assume to be occurring in the spring and the fall. But we know that we are seeing a lot more incidences of these under ice algal blooms in more places and at more times. And so one of the questions is, did we miss something before, or is this a new development that we just weren't looking for in the first place? So the objectives of this work is to understand what happens in lakes in the winter, what happens when nobody's looking, and then more importantly, does it matter? Are we seeing, is this worldwide expansion in summer harmful algal blooms associated with changes that have occurred in the seasons where we're not collecting data? And so we formed these thoughts and these hypotheses because nothing happens in the cold and the dark, right? Plants don't grow. We put things in our refrigerators to preserve them. And so it wasn't worth the effort to go out in the winter and collect things because nothing exciting was happening anyways. The other is more of a logistical problem. As you all probably are aware, it's very difficult to get out on frozen lakes, and as things change, it's dangerous in some cases when that ice cover is unsafe. And so we don't tend to go out and measure them for logistical reasons as well. But we know that if we get the right combination of light and nutrients, you can get these under ice phytoplankton blooms. And so, are these changes we're seeing a case of the more you look, the more you find, or is something changing? And so we know that winters are getting shorter. Predictions indicate that the percentage of ice-free winters will increase from 2 to 60% by the end of the current century. So that's some scary statistics for those of you that enjoy your winter sports. We did some comparisons. There was a global conglomeration of under ice scientists who took data from the summer and the winter and compared chlorophyll A concentrations. And so what we assumed to find was that winter would be negligible and we would see relatively high concentrations in the summer. But we were surprised to find that the under ice chlorophyll A concentrations were half that of summer. And so when you think about scale this up to kind of our year-round understanding, it's important that they're not zero, right? We're missing, half of what's happening in the year-round seasonal cycle. And so this then leads to questions. Are we seeing more harmful algal blooms as a result of things that are happening in the winter? And so I wanted to kind of open up the black box of the winter and shoulder seasons. And so by shoulder seasons, I'm referring to the spring and the fall, and ask, what is the year-round phenology of chlorophyll A concentrations and metabolism across a latitudinal gradient of lakes and reservoirs? And so the prediction would be that as we get these climate-induced increases in year-round light available to photosynthesis as a result of shorter winters and reduced snow and ice cover, will this result in increased algal biomass and autotrophy? And so we know in temperate systems we have two different kinds of lakes. If they're ice covered, we consider them to be dimictic, and so they have that layer of ice and snow on the top of them, which means that we call it inverse stratification. So instead of mixing throughout the water column, the phytoplankton are mixing in this top layer as a result of convective mixed layers. And so most of the biomass you'd expect to see kind of right under the ice layer there. In monomictic systems, which is what we have down in Missouri, we have kind of continuous mixing all year, or throughout the winter and shoulder and spring seasons. And so those phytoplankton populations are seeing, over a 24-hour basis, only a portion of that light as they circulate down throughout the water column. And given predictions about reductions in ice cover, it does beg the question, are we going to lose our dimictic lakes? And will, the current future in which Europe is already experiencing be these monomictic systems? And if so, how different do they function than what we're typically used to? And so we published some work last year looking at these convective mixed layers. So we observed that it was the snow on top of the ice that dictated how much light got through. And the light coming through can drive these mixed layers, so it can determine how much light the phytoplankton can see based on kind of circulation and physical patterns. We collected some data, some profiles of chlorophyll A concentrations, and found that they lined up pretty well. So when we calculated this convective mixed layer, as shown by the dotted lines there, you can see that the peak in chlorophyll A concentration falls kind of nicely within that realm. And so these convective mixed layers, how deep they are, how often they form, is going to be important in terms of what dictates phytoplankton biomass and productivity in the winter months. So I mentioned I was going to look at a latitudinal gradient. I did kind of a space for time approach here, where we looked at a variety of different Canadian lakes. So Lake Simcoe, located in Ontario, and then some Saskatchewan reservoirs. Compared this with some, as I mentioned, I inherited a long-term data set in Missouri, so that explains all the shapes in Missouri there. And then there's one that has been instrumented by the USGS in Kansas, a drinking water reservoir. As you can see, they represent a variety of different trophic status. So oligotrophic would be pretty clear. You can see pretty far down into them, whereas hypereutrophic and eutrophic would be kind of more greeny color and have a lot more nutrients in them. So we know that we have this gradient in trophic status and also an ice duration. So in Saskatchewan, you have an ice duration where you can drive, your pickup truck to sample on your meter thick ice for at least 5 months of the year, compared to Lake Simcoe, which has, less ice cover relative to the monomictic systems in the Midwest. And so we know that light is a master variable, as we heard earlier, about controlling plant growth. So often when we look at bottom-up processes affecting phytoplankton, we're looking at light, nutrients, both nitrogen, phosphorus, and temperature. But some work I've done and some understanding we have in the recent literature is during the winter and shoulder seasons, we have a lot of nutrients usually. So the nutrient limitation is not usually a problem during these seasons. And so really changes in light and temperature are going to promote algal growth. And so again, we know with these climate change predictions, we're expecting to see more light because you're going to have less snow, less ice, and maybe shorter convective mixing layers. So it's going to be spending more time in the light and increases in water temperature. The reason I can kind of quickly dismiss the nutrients from this talk is because we did do a variety of nutrient status indicators. So this is looking at physiological indicators, looking at how the algae respond to both phosphorus and nitrogen. And so we did a variety of different experiments like this, including some stoichiometry on a variety of these different ice and snow-covered systems in Canada, and found that in all cases they were sufficient. So for each of these indicators, there's a threshold to indicate deficiency or not, and so all of these were well above deficiency indicators. So they have enough nutrients in the winter, and so the focus now is going to be more on light. And one of the hypotheses to why they have so many nutrients is related to processes that occur under the ice. So as we get phytoplankton growing in these top layers, they're going to eventually die and sink down into the bottom layers through decomposition. This consumes oxygen. So we get changes in the redox conditions at the sediment-water interface. And this results in things that have been holding on tightly to phosphorus letting go. And so we see increases in phosphorus coming in from these bottom waters, which we refer to as internal phosphorus loading. And so I did some work in Lake Diefenbaker, which is located in Saskatchewan, and measured this internal phosphorus loading rate in the winter and the summer months, and found in that system that it was higher in the winter. In fact, 3 times higher in the winter. The oxygen concentrations were lower in the winter than they were in the summer, and we think this has a lot to do with it. And so again, this is another, internal phosphorus loading is another measurement that's not typically done in the winter. And so, begs the question, is this happening in more places, and could this be one of these primary sources of nutrients under ice? So the focus is now going to be on light, but before you can start looking at light and worrying about whether or not chlorophyll A concentrations represent phytoplankton biomass, you need to consider photoacclimation. And so as I mentioned at the beginning, chlorophyll A is a pigment that phytoplankton produce. Different types of phytoplankton produce different amounts of this pigment, and they can modify it based on the light conditions. And so under light deficiency, like we'd see in the winter, they would produce more chlorophyll A per biomass, and then under light sufficiency, which we tend to see in the summer in some cases, you would see the smaller ratio of chlorophyll A to phytoplankton biomass. So I first wanted to kind of correct for this or worry about this before we continued on with year-round analyses. And so here is the different types of reservoirs I worked on. I used different correlations to see whether or not chlorophyll A concentrations were correlated with biomass, so including microscopic counts of phytoplankton biovolumes, concentrations of particulate organic carbon, and particulate organic matter. And so we'd expect to see if there was an issue with this, that in the winter months you wouldn't see a strong correlation between the two. But I've got all checks there because they all checked out and correlated really strongly during these light deficient conditions. So I felt validated now in proceeding to look at year-round changes in chlorophyll A concentrations. So I'll start by looking off at the largest resolution and then scale down to some measurements that were taken on a daily basis. This is one that are dimictic with some serious ice cover. You can see here the chlorophyll A concentrations on the y-axis, and the x-axis is months. And so we don't see what we would expect to see in terms of that typical bimodal pattern. And so in fact, the maximum concentrations that were recorded under the ice were occurring outside of this kind of traditional sampling window. So if you had only been sampling in the summer, you'd be missing your pulses of production happening outside of that season. Moving now to Lake Simcoe, which is a mesotrophic lake located just north of the city of Toronto. We also did 4 years of year-round work on this system and found, again, the peaks and maximums in chlorophyll A concentrations were happening under the ice in winter. So again, outside of this traditional sampling window. In Lake Simcoe, it's a bit complicated because they do have these zebra mussels, which we know can improve the water quality or clear the water column. And so I mentioned kind of the difficulties of sampling in the winter. So one of the ways we overcame this, or the Ontario government overcame this, was the use of water intake pipes. So this is data that they've collected on a biweekly basis for decades from a water intake pipe located in Beaverton, just kind of in the eastern basin there of Lake Simcoe. And so when we compared the long-term data sets pre-Dreisenids, so those are the open symbols, versus after we know that Dreisenids invaded in 1995, we see significant differences. And so you can see here that on a year-round basis prior to Dreisenid mussels, we see that nice bimodal pattern that we would expect to see, but after they invaded, we saw this significant decline in phytoplankton biovolume in this case. And so, we're not sure if this is a result of decreased grazing in the winter months as a result of these Dreisenid mussels, because they are temperature dependent, or whether this is reflective of changing light conditions. But we noted that after Dreisenids were there, again, the maximum chlorophyll A concentrations are occurring under the ice in the winter months. So moving on now to polymictic and systems that do not have ice cover. This is the drinking water reservoir in Kansas. This has had issues in the winter months with taste and odor compounds in its drinking water. And you can see that they've got a wonderful 15-year long-term year-round data set. And again, it's noisy, but it's not that typical bimodal pattern that you expect to see. And the most significant highest concentrations of chlorophyll are occurring in the winter months. Moving on now to a variety of reservoirs in Missouri that span that trophic gradient. We have data sets from 1994 and 2004 where these were sampled on a biweekly basis. And so again, you see kind of this bimodal pattern, but it's been pushed and shifted with, again, the highest concentrations happening during those off-season months. This is another urban reservoir in Missouri that was sampled on a daily basis. And again, over a 4-year period, you can see kind of this crossing X pattern to the data set with, again, the highest concentrations occurring outside of the window. And so when I put all this data together and tried to summarize it and kind of figure out if there was any overlying patterns, I separated the data into duration of ice cover, so less than 70, 70 to 100, and then greater than 100. I normalized the data because it did represent a different trophic gradient, and so to kind of equalize or account for that, use z-scores here. And I indicated the different seasons by the shading. And so summer is the dark gray and winter is the light gray. And so what I found was that when ice cover duration was less than 100 days, the top two figures, the seasonal differences were significant, with the maximum chlorophyll occurring during the winter. In fact, it was twice as high as what we measured during the summer. When we used the data sets that had this long duration of ice cover, we saw no significant difference in season, which in itself is significant, right? You'd expect winter to be lower, and there was no difference over all of the months. So it appears that our traditional paradigm of this sampling season may need to be re-examined, and we need to maybe open up the shutters on the window and look at a broader year-round basis. And so one of the questions that comes from looking at chlorophyll concentrations is, does this correlate into productivity? And so what does this mean for carbon balance and whether lakes are sources or sinks? So we measured aerial gross production, AGP, for a lot of these systems and found this significant positive relationship with chlorophyll concentrations. But as you can see, the R-squared value there is not fantastic. So it's not always relating to production, but it is interesting when you look at metabolism. So this is looking at the ratio of production to respiration. And so if it's higher than 1, it would be considered autotrophic, and if it's less than 1, heterotrophic. And so again, the assumption would be that lakes under ice in the winter are considered to be heterotrophic. But we found that in our larger systems, so Diefenbaker and Lake Simcoe, we saw them go into this autotrophic range under ice during the winter. When we put this in perspective of a year-round basis, so comparing now open water, which are the circles, to the X's, which are under ice, you can see the relationship in the open water season between production and respiration, but you can see during the winter months, it's mostly dominated by respiration. And so anything above that line is considered to be autotrophic, and anything below the line is heterotrophic. And so you can see that the open water gross production was much higher, right? So 17 to 66 times the under ice rate. The open water respiration was 8 times over the under ice rate. So, we talk about these chlorophyll peaks and things like that. Does it correlate into production? Not significantly, but still we get some incidences where we're pushing into that autotrophic conditions. And so the reason we assume heterotrophy and that nothing happens under lakes in winter is a lot to do with light, right? It's dark, nothing grows. But of course, this light environment depends on the depth of the snow and the ice. And so I calculated the light accounting for these convective mixed layers. So this is E24, which is a calculation of how much time a phytoplankton cell spends in the water column. And so it accounts for the attenuation coefficient, so things that are attenuating light throughout the water column, as well as the incident irradiance to account for daylight, and this convective mixed layers. And so what we found in Lake Simcoe was that there was no significant seasonal differences in mean light. So light was not significantly lower in winter. And in fact, again, you can see that the highest occurrences of light are occurring right before the ice comes off. So ice cover is shown by that blue horizontal line, and you can see right at that point where ice is coming off, we see these large pulses in light, which relates to these large pulses in chlorophyll concentrations. And just to see if Lake Simcoe was weird, I mean, it does have zebra mussels after all, I compared this with Lake Diefenbaker, which does not. And again, calculated this E24 value. Ice cover is a little bit longer in Saskatchewan, so that April point is under ice. But there was no significant seasonal differences. So again, not lower light in winter. So what does this mean? How low can you go? How little light can they do anything with? And so here I'm showing photosynthetically active radiation, or PAR, related to snow depth. And so we used some threshold from the oceanographic literature to determine how much light they need for biomass accrual, which is the top dotted line, and for photosynthesis, which is the bottom line. And looked at a variety of these different systems and found that 57% of the time, they were above this limitation for biomass accrual. So they had enough light 57% of the time. They were limited below that bottom threshold about 32% of the time. So what did this mean in terms of snow depth? Could we come up with a number where we saw this shift into this light limitation range? And so some literature from Lake Baikal suggests about 10 centimeters is this point of snow. And when we did our calculations, it was 13.5 centimeters. So it appears that when you have more than 13.5 centimeters of snow, they're pushed into that light limitation range. So then to kind of experimentally manipulate that, we did some snow removal experiments where we measured light under the ice and snow, then we shoveled off the snow, and then we measured it again to see what difference the snow made. And so the snow cover is the red, and then the blue is the no snow cover, again over this gradient in snow depth. And so we saw differences from what we saw before when there was snow was present. So now only 14% of the samples were light limited for biomass accrual and only 7% for photosynthesis. So removing the snow resulted in a 40% increase in light available for photosynthesis and to produce biomass. So then one of the thresholds we kind of like to ask is, how much can you take, right? If we gave those low light adapted phytoplankton more light, would they be able to do anything with it? Would they be able to handle it? And so for that, we use the EK parameter from PE curves as a light saturation proxy. And so you can see here that during under ice versus during open water, we're well above these thresholds. So again, this is the field conditions of what light was like under ice with snow, open water, and under ice with no snow. And you can see that while open water, obviously has a higher EK value, they're still well above these thresholds in terms of the winter values. So it appears that if more light was available to them, they would respond and you would see an increase in photosynthesis. So it appears there is this interactive control of both light and nutrients on these under ice phytoplankton populations. We found that a 6% increase in this under ice light transmission, which corresponds to about a 5 centimeter reduction in snow, can cause a 6 times increase in primary productivity. So significant changes are happening when you increase light. We know that they're well adapted to these low light conditions, though they will respond to increased light and can result in these production pulses. And so we often assume that during the winter, light is the control on under ice algae, and they're sitting down here where that yellow star is in the light deficient range in terms of the relationship between light and phytoplankton productivity. But the question is, as we get improvements in light conditions as a result of climate change, are we going to push them up into nutrient control again? And if that's the case, then we need to be a little more concerned about what mechanisms are happening under ice to control these nutrients. And so what happens in lakes in the winter? Well, we know that there is low under ice light, but mostly due to snow cover and not as much to the depth or thickness of the ice. We know that they're light limited, but they can respond to increases in light. We know that they have a lot of nitrogen and phosphorus, and so as soon as they get that pulse or that boost of light, they have all the other resources they need to take off, and this may be related to internal nutrient loading. And although we see these under ice peaks in phytoplankton, it's mostly just response in terms of biomass and not necessarily in terms of productivity. Though autotrophy can occur under ice. And so when we do these kind of carbon balances and accounting for different things, we need to also account for what's happening under those, in those winter conditions. And so the question is, are we in fact seeing shifting peaks, or are we just looking for them more? Is it a consequence of these climate-induced shorter winters and improvement in these light conditions resulting in changes in the convective mix layer? And as we move forward and potentially lose all our dimictic systems, what does that look like if we all had monomictic conditions? So what are we going to expect to see, and how can we try to maybe mitigate some of that? So I think it's important to understand both winter and shoulder season dynamics as we try to predict future changes in year-round lake function. And so are we missing these pieces of the puzzle? And with that, I'll thank you for your attention and welcome any questions you might have.
There you are. So it's time for questions. Oh, Warwick, yeah.
Well, just to break the ice.
Sure.
Thanks there, Rebecca. That was really great. And of course, makes a lot of sense to us working up in the north, where very often we have continuous ice throughout the year, and we have fish in lakes.
Still, yeah.
So clearly that's not spontaneous generation. There is a food web. And your arguments about light also make a lot of sense. I was just wondering whether there might be some additional variables also that are kind of superimposed. For example, well, in our situation, day length is critically important, ultimately going up to a day length of 0
right.
as we head northwards. Do you think that could have an influence in your gradient, for example, as you go from the lower states up to the border with Canada?
Certainly, yeah. Those E24 calculations that I showed did account for incident irradiance over a 24-hour period. So, they would capture those daylight changes, over that latitudinal gradient. But, yeah, whether or not...
Is that a big change over that gradient, or is it not enough of a latitude to really have an effect?
I would assume there is. I don't recall. Yeah. But, yes, there must be, right? So, yeah, that's going to factor into that light. But it still, wasn't enough to bring... because of those convective mixed layers, the mixing dynamics seem to keep them suspended up in that light when it is available. And so, yes, it would factor into it.
So, of course, another variable that's changing throughout the year is temperature.
Yeah.
And the biomass at any particular instant of phytoplankton is that balance of gains and losses. And one of those loss processes is zooplankton grazing. Mm-hmm. Do you think there could be any suppression of top-down effects in winter that could contribute towards the persistence?
Yeah, that was certainly our story on Lake Simcoe with the zebra mussels. When we looked at that before-after data, it became pretty obvious that was the impact those were having in terms of grazing.
As benthic grazers.
Yeah. As benthic grazers, but they do have access to the whole water column too during that point in time. So yeah, they would have that impact. And the other caveat of that study, it was an intake pipe. So it was actually located in the nearshore area of the lake. It was about a 7-meter depth. So they would maybe have more access and more effect there than it would have if we'd had that data from the offshore areas of the lake, because Lake Simcoe is like 45 meters deep. But yeah, in terms of zooplankton grazing, we assume that they would be temperature dependent, and so you'd see reduced grazing weights in the winter. But some of the work in the ecology letters paper by Stephanie Hampton et al. Showed that zooplankton, were as active. They were not zero either under ice in the winter.
And there's no evidence of them running down over the course of winter.
I don't think I've never seen that data. So most of these monitoring programs, like Wisconsin has an LTER, things like that, they go out 3-4 times in the winter and collect data, and that's, probably the basis of most of that data set. I haven't seen anyone do zooplankton work on that resolved basis. I know Ted Ozerski at the University of Minnesota is interested in those questions, and I think he's got some work following up on that, but I haven't seen the results.
Okay. And just finally, one other variable you probably thought about is bright light effects and UV radiation exposure.
Yeah.
That also is going to be changing with climate change. Do you think that's a factor, particularly as you get into that spring period, your shoulder period, because you're getting close to the summer solstice and potentially maximum UV irradiance?
Yes. Yeah, definitely it's going to play a role. And I didn't collect that data. We didn't have, the good light cores and stuff like that would measure light in that range. So no, I imagine it would have an effect. Not in Missouri. Our water there is relatively brown, so we kind of we dismiss it fairly readily in those systems. But in the more northern systems, the Canadian systems, that would play a role. Yeah.
Thank you.
Yeah. Thank you.
Thank you, Warwick. And any other questions?
Everybody wants to go home?
Well, it's 5 o'clock. I know people are a little bit tired now. Okay, Jonathan, you still have a question? Yeah, go ahead.
With the rise of biomass, because of the rise of concentration of nitrogen and phosphorus, you can have a rise of anoxia too in the bottom of the lake. Mm-hmm. Does it impact the zebra mussel population a lot?
Yeah, it's an excellent question. So in Lake Simcoe and things like that, it doesn't go hypoxic in the bottom waters where we have the Dracaenid mussels, and so I think that's why they've been so effective in Lake Simcoe and the Laurentian Great Lakes and things like that. But I'm learning now in Missouri where I am, our hypolimniums do go anoxic for pretty much the entire summer. And the before and after data that we've looked at so far to see their impact, so looking at total phosphorus, total nitrogen, secundus depths, things like that, there's no change over time after their invasion. And so that might be one of those factors. So the theories around that are, well, the water temperatures also in the summer months can reach like 35, 40 degrees Celsius. So maybe it's too warm for them to do well in those southerly climates. Maybe because of this high turbidity, their siphons are getting full, so they can't graze as effectively, and then maybe they're getting asphyxiated. So future work. So right now we know that there's Dracaenids in those lakes because people complained. So we don't have any kind of density or distribution or anything like that. We just know that somebody mentioned they saw, well, it was confirmed by the government, but just, one or two. And so we need to go back now and try to map them out and see if we can correlate it with, these areas of anoxia and see why they're not having an impact, which could be interesting, So as they move farther south and farther west, if these types of systems don't show a large impact, then maybe, it's not the panic button that was hit kind of around the Laurentian Great Lakes and things like that.
Excellent. Any other questions?
Well, we just have to ask you about cyanobacterial blooms in Missouri. Are they on the increase, as we're seeing, for example, in southern Quebec?
If we had long-term data, we could answer that question.
So like Jack's data?
No, we started doing it a couple years ago. So we have 3 years of data, which I'd hate to talk about trends with 3 years of data. What we do know in Missouri is that we have lower concentrations than the surrounding states. Mind you, we're surrounded by Iowa and Kansas, and this is the Midwest. So we can't talk about long-term changes, but we are starting to look at different types of toxins. And so microcystin is, we did that for the 3 years, and that's the data set we have. But when we started looking at phytoplankton samples, we have different species that produce different types of toxins. And so this year we actually convinced the state government to fund us for the 4 toxins. So now we're looking at cylindrospermopsin, anatoxin, and saxitoxin. And we did see some incidences in June of really high cylindrospermopsin concentrations. And so maybe the low concentrations were just the wrong type of toxin, because blooms are abundant. You can visibly see them from the surface a lot.
So is there any anecdotal information that you can get from talking to people about their grandmothers, for example?
Yeah, so we're starting to work with the vet college because they have a lot of incidences of cattle death, which they attribute to harmful algal blooms because the lake was green. And so, we're trying to work with them. So with this citizen science program, we're pretty set up to provide people with kits, right? So, here's the kits, here's the protocol, collect the toxin sample. And so we're trying to involve vets in this as well to be able to get that kind of evidence that it in fact was a harmful algal bloom that caused the cattle death. Right now it's anecdotal and green lake, dead cow, must be. And so we're trying to get some evidence that is the case and put those numbers on the books.
Okay. It would be interesting talking to farming families and ask them, is there any kind of historical evidence that there were these kinds of problems in the past?
Yeah, it's interesting. On Lake Diefenbaker, when we started working there, we started asking the public, we started there with the social science component, what are the issues? And they all said water quality's declined. My grandfather used to be able to see to the bottom of the lake, now he can't see. And we did all kinds of work. There was a special issue in the Journal of Great Lakes Research where we used 4 different approaches. We used satellite imagery to go back in time and look at surface algae. We used paleo approaches, and we used the kind of long-term governmental monitoring, and there had been no significant long-term change in water quality. But people's perceptions were that it had declined. It's often, disconnected, or we're paying more attention too, is the other case. So it's hard, yeah.
Yeah. No question? So in aquatic ecology, do you look also at CO2 or just CO2?
Do we, as a greenhouse gas?
Yeah. How it can influence the algae bloom?
Oh, whether or not they're carbon limited? Mm-hmm. Is that? Yeah, there's work on that. I mean, that was certainly a lot of the work in the 80s really was focused on whether or not carbon limitation occurred. And when you get these high density surface blooms, that can certainly be a thing. There's been work in Iowa recently. John Downing group has looked at that and found, and so ones with carbon concentrating mechanisms are getting the advantage over the other types of cyanobacteria, and those happen to also be the toxin producers. That has a role to play as well. I personally don't have any carbon data, but I know some people are working on that.
Okay. So then, once again, thank you very much for your nice talk.
Thank you.
We have also a little gift for you, Rebecca. So it's exactly the same. So you won't be fighting to each other. Okay. Yeah, great, excellent. So. Je vous remercie tous d'être venus à participer à ces 2 conférences. En fait, c'est notre première conférence du CEF, Centre d'études de la forêt. Et nous avons notre directeur présent ici, professeur Louis Bernier, qui est sur place. Et je voudrais remercier aussi Warwick, qui a certainement fait un bon travail pour amener les gens de son groupe et du centre eau. Je crois qu'il y a plusieurs participants, membres du centre eau ici. Et évidemment, cette conférence n'aurait pas pu avoir lieu, évidemment, sans l'invitation. Tout a été organisé par le ministère des Relations internationales de la francophonie, qui est représenté ici par Renaud Maud, qui est en charge du dossier États-Unis-Québec. Et évidemment, Girafe, professeur Alain Olivier, qui est la chaire, qui est titulaire de la chaire en développement international, et le CEF. Donc nous avons tous contribué à faire de cet événement un succès. Vous avez derrière vous les personnes qui sont les artisans du CEF. Natacha Fontaine, qui a fait un travail formidable pour diffuser l'information, et notre ami Pierre, qui est là, qui est en train de nous filmer tous. Donc, je tiens tous à vous remercier pour cette belle conférence. Je crois que ça a été un grand succès pour ma part. Et merci d'être venu en si grand nombre. Je vous remercie. Voilà.